Hello, dear aquarist, dear fish keeper or breeder. I think you want to know more about Tetrahymena. Yes, the terrible parasite that makes severe wounds and lesions, also called the guppy killer. Well, I will discuss two studies that I found and I give you my findings, my opinions as a fish doctor with the experience I have dealing with this parasitic infection, and of course giving you suggestions what you can do about it as a treatment. At the same time, I will give you the links below where you can also find those studies. And the first study is about the impact of three different fish medications or drugs on tetrahymenosis, or diseases or infections caused by Tetrahymena. And they were using environmental DNA techniques. Well, this is a technique to check with DNA. I will show it also in the next study to find out the parasite and the bacteria in the water. Well, the paper particularly discusses the relationship between microbial diversity and fish mortality. Well, we know when there are too much microbes in the water, there is a big risk that the fish gets diseased due to weakening immune system and more proneable to get a parasitic or bacterial infection. And they revealed the pathological damage caused by the parasites Tetrahymena on goldfish. And they discussed the different manifestations of three drugs that's been used in the treatment. And they find out that the application of Nitrofural had a significant effect on the treatment. So this is an antibacterial medication. So I give you the abstract and then I will conclude my findings on here. Because Tetrahymena will cause severe economic losses in the industry, and there is still lack of study to use specific drugs on the prevention and the treatment. And here they did an experiment with three medications. That means Coppersulphate, Nitrofural, and methylene blue in combating the Tetrahymena in goldfish. They used the eDNA sequencing and PCR technology to extract the environmental samples. And I go more in detail in the next study on the bacteria which can be associated, can be found with Tetrahymena. But what's interesting in this study here, that their findings revealed that when they used the drugs, that Nitrofural had the most promising effects, followed by methylene blue, and then copper sulfate had very little effect. Why? Well, because copper sulfate is particularly working to control parasitic infections like oedenium or controlling of algae growth. Methylene blue is a general broad spectrum antibacterial, most likely. And nitrofural, well, in the study proved to be working to control tetrahymenosis or tetrahymena disease, but in my opinion, it controlled the bacteria. It controlled the bacteria which usually come together, infect together with the parasites, the wounds, the lesions, and kills the fish. And also that use of that product nitrofural also controlled the bacteria in the water. So it cleared up the water, the negative effect of the bacteria. And at the same time, when you control the bacteria, and in my findings, I've been using antibiotics when we treat Tetrahymena because the wounds are infected with bacteria. And if you don't heal the wound, well, the parasite remains to have a big chance to keep on damage. Of course, trying to treat Tetrahymena is still a challenge. And I usually use for that formalin and malachite green combined with salt. But in the other study that I will show, I conclude about this one of my findings, and the other study shows other things you can do, and that's I want to share with you the next study. The next study is a little bit different and did some other studies, and I highlighted in that study the, how can I say, the most important matters that concerns us. I will go from the beginning. It's the interaction between Tetrahymena and bacteria under different physicochemical conditions in guppies. And they were using an eDNA method, where this is a laboratory method. They can check the parasites and the bacteria in the water with this method. Very practical in large aquaculture facilities when you have the tools available. This is new technology. I will not go in detail about those technologies today. I will particularly talk to you about, well, what was the basic important findings here, because they were looking for the relationship between Tetrahymena, the bacteria, and the guppies under different temperatures and different pH conditions. So the abundance of Tetrahymena in water and the relative abundance of bacterial species and the pathological observations were studied. And the results of the study showed that several bacteria were related to Tetrahymena and that environmental factors affect the interaction between the bacteria and the Tetrahymena. So that is the basic, there is a relationship between bacteria and parasites in this case. Like I also talked about or showed before, that like gill flukes or skin flukes also have kind of bacteria with them that accompany that parasite and also make wounds combined to the parasites. And here it's the same thing together with other factors. Because, well, those protozoa cause severe damage, severe losses in our industry, particularly the guppies, it's well known. And the results, well, showed in detail that the bacterial communities of water samples were mainly composed of species assigned to proteobacteria and bacteroidetes and other bacteria. I will not give all the names, you can read it for yourself, that were positive correlated with fish mortality. And the Tetrahymena also was abundant available together with a temperature. The temperature played an important role. Also, other bacteria played a role in different groups and were had an effect with different environmental factors that affect the interaction between the bacteria and the Tetrahymena, and in particularly lower temperatures and higher pH were more beneficial for preventing disease outbreaks. So that's something to think about when you deal with guppies. Higher pH, of course. Better to get a little bit more salt in the water, keep it a little bit light, brackish water. That's what guppies genetically are preferred to live with. And lower temperatures, because lower temperatures is usually giving a bigger chance for bacteria to grow. So to study, I will give you the most important facts about the materials, the methods. You can read it for yourself. So they had guppies set up in a 2-liter tank with 10 guppies, and they were fed, well, not fed during the treatment, they didn't got anything. They were small guppies, well, approximately 10 grams, 4 centimeter, a normal sized guppy. And then there were the ones which have to be tested for the infection. They were treated with 10,000 Tetrahymena parasites were added to the water for the experiment. And then there was a control group. And then there was, well, different pH levels were set, 6.5, 7, and 7.5. That was the pH. And, well, they used for separating, the cells, the parasites. They collected it from the gills and from the mucus of the infected fish, so they could study them. Well, I'm not going detail in the technicals, the DNA extraction. Those things you can read if you're interested. Uh, I will go particularly here. What interesting is that The data from the 30 samples, including the 3 replicates, each of the control and the experimental groups under treatment of several different temperatures of 20 degrees and 26 degrees. So that was checked for, and at the 3 different pH levels, 6.5, 7, and 7.5, that were analyzed. Well, we know that the guppies cause severe damage, muscle tissue, skin, wounds, and spread throughout our body. So here they show you the pictures, which of course I show in my videos. Here we see a healthy guppy, no wounds, but here we see another guppies which are badly damaged. You see the lesions, the open wounds. You see the bad tails, which are really falling apart. You see the cloudy eyes, the big eyes, cloudy pop eyes. You see here more whitish lesions. You see gills which were infected. And then they show here the microscopic findings of the parasites and of the tissues where they collected the parasites. Also internally, on the left, there is A, D, and A is the healthy liver. Even the liver had some, how can I say, some vacuole openings. The gills here on the left, D, is a healthy gill, but E and F were badly affected gills by the parasites. And then, of course, there is the muscular tissue. Well, on the right, H and I here were badly affected muscular tissues showing in examination in the laboratory. Well, these are the findings which we know when we deal with guppies, what kind of lesions they have. So I continue further on that, well, the different water temperatures were used, and then they have here the table, and that is interesting to show. It's the mortality rate. Here it's 20 degrees on the left, and on the right it's 26 degrees. So you can see 26 degrees is a much higher, nearly a double amount of mortalities in 26 degrees than 20 degrees. I was surprised to see that even the control group, which did not have Tetrahymena, also had a mortality of nearly 25%. Well, I'm questioning, how could that be? And then due with the pH, you can see with a lower pH here, 6.5, it's a higher mortality in comparison to 7.5. It's half the mortality. Even still, mortality rates were recorded with the control group. So there was also a diversity in the flora, the microflora, the bacteria, and I go in detail here at the end. You can see it here that with the temperature, there were higher amounts of bacteria at higher temperatures. This is a fact we know. Bacteria do well. And depending on the pH, there was different levels of bacteria available. Of course, pH and temperature controls the bacteria you can have in your system, in your aquarium, or in your facility as a breeder. I don't go in details all about the bacteria. You can read about it. It's very heavy stuff. For me, it's interesting for you to tell what's important I found. And of course, the conclusion in the discussion says that there was an interaction between the protozoa, Tetrahymena, and the bacteria that play an important role. The ciliates may change their reproductive system and their feeding behavior according to the abundance and the diversity of bacterial microbiota in the environment. That means when you have a dirty fish tank, too much food, too much bacteria, too much chance given to the parasites Tetrahymena to do well. And that's the thing what happens when you have Tetrahymena outbreaks. Of course, that you create opportunities for Tetrahymena to do well when there's a lot of bacteria in the water, a lot of pollution, dirty water. And environmental factors also play a role, like the pH and the temperature. It is known for a fact, and that was studied here in this study, that the setup was made with the pH and the temperature. And in the 26 degrees, higher temperature group, more favorable conditions for the parasites, with you causing a higher mortality. And the same thing counts for the pH. When the lower pH, the higher the mortality of the guppies. So things to learn about that. Those results showed, well, there is some factors playing a role, not only the bacteria, but also the physio and chemical factors playing a role. And that you should think about when you culture fish, or keep fish in breeding tanks, that you keep it clean. So there is an interaction that aquarium owners know that increase the pH using salt, keep them light salty water, and adding probiotics is a suggestion to be made to make the fish immune, improved situation, and use cooling. Too hot water, more than 26, or I always say above 24 degrees, is a big risk of having this problem. And that talks about the bacteria which are correlated with the temperature. Rhodobacteriaceae, call it all names. Also, they talk about Flavobacterium, and Flavobacterium is columnaris. And columnaris was seen not to be positive correlated, and they presumably that those Flavobacterium, here it says, they are pathogenic to the freshwater fish, and they cause, of course, serious infections. I have different videos on that. But in this study, it was negatively correlated with the mortality. And it's interesting to know that this might be due to the interaction between the Tetrahymena and these bacteria. Because it may be due to the phagocytosis, it's also possible that the temperature and the Tetrahymena affect them together. The bacteria may become inactivated and consumed by the ciliates in the environment. So the Tetrahymena are potentially consuming Flavobacterium or the columnaris disease. So these are things you can consider, that some bacteria might develop and some don't develop. And to be honest, I have seen honestly not too much Flavobacterium or columnaris together with Tetrahymena infections. It's usually a mix of bacteria that can appear depending on the bacteria which are available in the water. So the conclusions was that certain bacteria exhibited an inverse correlation with fish mortality, and the abundance of Tetrahymena under different conditions, like he said, lower pH, higher temperatures, demonstrate the relationship between bacteria and Tetrahymena, and that could be influenced by environmental factors, things you can do something about. So that's the main point I want to make. You can do something about making your fish or keeping your fish healthy. Guppies, particularly for guppies. More salt, higher pH, lower temperature. And probiotics, like we do with our Dr. Bassleer biofish food, like the Forte, helping to improve immunity of the fish. So this study explained for me, or my findings that I always say, with the holistic view, combination bacteria in the water, pollution in the water, high temperatures, not good pH, affects the health of the fish and giving a chance to the parasites like Tetrahymena to do well. Any comments? Any questions? Or you want to learn more? Subscribe. Thank you for watching.